Without further ado, I'd like to introduce to everybody our final keynote speaker for this evening, which is Dr. Andrew Ha. Dr. Andrew is the Senior Manager of Deogen. He has a PhD from the University of Stanford. He strives to advance Deogen into the international market by accelerating global business plans and spearheading collaborative partnerships. Dr. Andrew, it's a pleasure to have you with us today. Feel free to provide a little bit more in depth of an intro for yourself. I was curious to see what you had your PhD in, and absolutely, it would be lovely to hear your comments on today's session.
Yeah, thank you so much for the introduction. Thank you so much. I did my PhD in stem cell biology at Stanford. I was also a scientist at Genex Therapeutic, which studies immunotherapies. And after that, I joined Deogen, which I'm going to talk about today. Let me, all right, can you guys see the screen?
Yeah.
Oh, perfect. All right, so I'll be introducing our AI platform, which utilizes the protein amino acid sequence to identify novel drug candidates. And this approach opens up a great potential for AI-driven drug discovery and development by bypassing the need for 3D structure. All right, so today's talk is divided into 3 sections. The first 2 sections are devoted to presenting our sequence-based drug discovery platform. Next, I will introduce our company structure. So I want to start off this talk by giving you one example of how Deogen's platform identified remdesivir as a potential drug for COVID-19. Remdesivir was approved by the FDA as the first treatment for COVID-19 in July 2020. However, Deogen was able to predict and publish a paper in January 2020, even before the 3D structure of SARS-CoV-2 was identified. And how do we predict this drug candidate without even knowing the structural information? The answer is we made this discovery by taking an unconventional approach. So instead of relying on structural-based approach, which is commonly used by most AI companies in the drug development space, we use the amino acid sequence of the target protein to predict drug candidates more quickly and efficiently. And why is that important? Approximately only 8% of total human proteins has been crystallized, and drug targets from AI-biotech companies that take a structural-based approach are restricted to this 8%. In contrast, the amino acid sequence of most proteins are already known. This means that 92% of human proteins that could serve as potential targets are overlooked. So our hope is that by utilizing the protein primary sequence, we can tackle this unexplored 92% in the drug discovery space and increase our chances of identifying novel drug candidates. Deogen's innovative technology has been recognized by a top global report, and Deogen has been selected as one of the top 33 leading AI companies in drug development for 2 consecutive years. Unlike most AI companies in this space, we have an end-to-end platform that supports not only the initial stages of a study, such as from target discovery to hit identification, but also lead optimization and preclinical development. And all of these advantages derive from our core AI technology shown here. For example, we integrated transformer model into our platform, which is used primarily in the field of natural language processing, but we are the first to apply it to the field of drug discovery. In addition, our platform includes a wide range of deep learning and machine learning algorithms, and these methods are used for target selection and drug candidate design. In addition to these advancements, we also have additional components that are unique to Deogen's platform, which support the prediction of binding affinity, binding site, and determines the prediction of uncertainty. I would like to briefly go over our 3 major platforms. First is DeerTrends, which is used for target discovery. We have developed a neural network-based feature selection algorithm called WX. Once we input publicly or privately available transcriptomics, such as NGS or microarray data, our platform can rank genes based on their discriminative index score, and which then can be used to select an optimal set of genes for target or biomarker discovery. Next, DTI is our drug target interaction platform that is used to identify hit molecules. Once we input protein and compound information, our platform can predict and provide binding affinity scores, as well as efficacy scores that is used for hit discovery. What I'd like to reiterate about our drug target interaction platform is that it is a sequence-based approach. On the left shows the type of input that we utilize. For small molecules, we use SMILES, which represents chemical compounds in the 1D string format. For proteins of interest, we use the amino acid sequence, which is also a 1D string format. Most AI companies requiring the 3D structure as the input are limited to candidates with a tertiary structure. However, Deergen's sequence-based approach bypasses this limitation. The right side shows the type of output that can be generated. Deergen's platform provides the binding site and binding affinity, as well as functional activity scores such as IC50 and EC50. Lastly, MolEQ is our lead optimization platform. Once we have a seed molecule, we can optimize the molecule until a desired threshold is achieved through a reinforcement learning process. In addition, our platform predicts both a final compound and the reaction pathway based on available chemical building blocks. Our platform is unique in that it can optimize substructures of the molecule while keeping certain part of the molecule the same. And what's important to note is that our model considers multiple properties at once and optimize the compound with differentially weighted parameters of interest, which is critical for tailoring the best compound for specific needs. Now here is an overview of our operating system with a flow chart of each of the platforms that I just described to maximize our AI learning capability in the drug development process. We have recently also established our own R&D center so that we can actively pursue targets identified in-house. And by performing wet lab validations on our identified compounds, we generate more data that can be fed back into our platform. And this creates a closed loop learning system that allows data from experiments to fuel future design. And this continuous reinforcement loop drives the perpetual growth in the power of our predictive models. I'd like to share with you two case studies. In the first example, we are targeting Alzheimer's disease. We use our drug target interaction platform, which uses target amino acid sequence, and in this case, FDA-approved drug libraries as inputs. Within 2 weeks, we were able to identify hits, and our experimental validation of the predictions looked very promising. Here, you can see the predicted and experimental EC50 values look very similar. The second case study shows one of our oncology projects. Here we have identified novel molecules for PROTAC or proteolysis targeting chimera, which utilizes targeted protein degradation as a mechanism for cancer therapy. We were again able to identify hit molecules in 2 weeks, and the results show a very potent anti-cancer effect. So for the last part of my talk, I would like to briefly give you an overview of our company. From our founding in 2016 until 2022, we have shown consistent progress and established effective partnerships with leading pharmaceutical companies and academic institutions globally. Today, we have over 20 projects in collaboration with 10 partners, and we believe that our R&D center will spearhead active expansion of in-house pipelines. We also established a site in the US to strengthen external collaborations. Currently, we have 4 sites, 3 in South Korea and 1 in the US. And to give you some context of our team credentials, we have equal representation in AI technology and life sciences. Currently, we have over 50 full-time employees that are experts in diverse fields. Our leadership board consists of leaders with 20 plus years of experience in AI and biological sciences, as well as business. And lastly, the following table summarizes our co-development and in-house pipeline programs. Our co-development programs are pursuing indications in various fields, including oncology, NASH, and CNS. Our in-house pipeline is currently in development, and we look forward to sharing details of our progress in the coming years. And that's all I have for today. With that, thank you for listening, and I'll be happy to take any questions.
Thank you so much, Dr. Ha, for your presentation. It was very, very informative. I think we have certainly got quite a few comments and questions relating to what you've discussed.